Let's talk about proteins. Proteins are essential to our bodies as they have many functions and are coded for by our DNA. In this video, I will explain the journey from the initial amino acid sequence synthesized to a functional protein. Amino acids are the monomers that make up proteins and consist of a middle carbon, an amino group, and a carboxyl group, and these determine what the N and C terminus of a protein are. Additionally, each has a side chain, commonly referred to as the R group. These side chains can vary, creating the differences between the 20 unique amino acids coded for by our DNA. Once the cell goes through the processes of transcription and translation in order to synthesize an amino acid sequence, the resulting polypeptide chain still needs to be folded. This is because the chain created is one-dimensional and non-functional, and in biology, shape determines function. So, in order to become functional, for example, break down a substrate or bind to a signal molecule, we need to change the shape of the sequence through interactions between the amino acids. When it comes to folding, there are four main protein structures. The first is the primary structure, which is just simply the amino acid sequence. There are no folds here and the protein is not functional. The next structure, the secondary one, is comprised of interactions between the backbone, the NCC, of the amino acids. The two main types of formations here are alpha helices and beta pleated sheets, both of which are formed by hydrogen bonds between an amino and a carboxyl group. An alpha helix is formed when an amino group of one amino acid bonds to the carboxyl group of another 3 or 4 monomers earlier in the chain, which creates a spiral shape. Beta pleated sheets, on the other hand, are made up of at least 2 or 3 strands of the polypeptide chain, which are bonded together, and can be done so in either parallel or anti-parallel fashion, depending on orientation. Then we have the tertiary protein structure. This is formed by interactions among the R groups of the proteins through hydrogen bonds, ionic bonds, disulfide bridges, or hydrophobic and hydrophilic interactions. Here, the protein folds even more, and in some cases becomes functional. However, many other proteins require the final structure. This would be the quaternary structure. Here, multiple polypeptide chains with tertiary structures bond and fold through the interactions listed previously. An important protein in our body that contains a quaternary structure is hemoglobin, for example. Folding is now complete, but how is it done? Protein folding occurs in the endoplasmic reticulum of a cell, and it occurs spontaneously and often begins while the C terminus is still being synthesized. Folding happens under very specific conditions to avoid denaturation, which can be facilitated through molecular chaperones, for example, which are also proteins. Now, there are other modifications that must be made in order to finalize the protein, but that is for another day. Thanks for watching.